Okay, very good evening, my dear friends. Welcome to today's session. Today's our resource person, Dr. THIRUNAHARI UGANDHAR sir, Associate Professor, Department of Botany, Kakatiya Government College Autonomous, Hanmakonda. Sir is going to explain about the most important topic in molecular biology or cell biology, that is meiosis. That is also we can call as reduction division, which is most important topic for all the competitive exams related to botany, zoology, microbiology, biotechnology, biochemistry, and also the competitive exams related to CPGET, CUET, APSET, TGSET, CSIRNet, NTA, CSIRNet, ARSNet, and also it is most important related to all plant sciences and including life sciences. So, in this regard, we would like to invite today's resource person, Dr. Thirunahari Yugandhar sir. Please welcome, sir, and start your session, sir. Please welcome. Please unmute yourself.
Sir, okay.
Welcome, sir.
So now starting the meiosis. This meiosis is also reduction division. So this figure has only cell division. The type of the cell division is G2, G1. That is called a mitosis. So this mitosis and also meiosis both are called cell divisions. So this cell division, first cell division is also meiosis, also called a reduction division. So this reduction division is which can take place in the plants, in the sporosporangium, microsporangium, megasporangium, or macrosporangium. The microsporogenesis, which formation of the microspores or pollen grains. Then megasporogenesis or macrosporogenesis, which is responsible to the four applied macrospores. So this type of the microspores are formation of the life, means applied gametes formation in the living organism of higher plants, of angiosperms, gymnosperms, and also living human beings and also other animals, which is also important for the life formation. So so the meiosis was first discovered and described in sea urchin eggs in 1876 by the German biologist Oskar Herdewig. Later in 1883, the Belgian zoologist Eduard van Benden further described it at the chromosomal level in roundworm eggs. So the roundworm eggs also important of the eggs formation. And the ovaries and also called a testis. These both are called a male and female reproductive organisms, which responsible to formation of the sperms and ova or eggs. That first division is called a meiosis. Majorly, what are the differentiation of the mitosis and meiosis? Major, that not completely. The meiosis and at a purpose. So the meiosis has a purpose of reproduction. The mitosis is a purpose of growth and development and repair. So that the mitosis is only division one, only mitosis one, not only mitosis two, but meiosis 1 and 2. The two divisions are there. So in the mitosis parental cell, which can take place in a diploid or sporophytic plants, also called diploid organisms, or 2N state. So the meiosis, the only which can take place in a diploid sporogenous cells and which reproduce which undergoes meiosis to form a 4 cells. Then mitosis daughter cells, only 2 cells are formation identically to the parental cell, but meiosis in only 4 cells, which are a formation of parental cell called the gametes germ cells or sex cells. But mitosis takes place in the somatic cells. The meiosis takes place in the reproductive cells, also called a also called as sporogenous tissues or sporogenous or sporangium tissues. These sporangium tissues which can formation in the sporangium of female or male gametangium. So this is also what are the mitosis and meiosis takes place here. That they also figures. The chromosomes are counting. The mitosis 46. The meiosis, which are also called the 23, or two pairs of the chromosomes are there. Meiosis. The meiosis is also gametes formation applied 23 also. This is the one type of the reactions of meiosis are first gametes formation. But here is also the meiosis are then anaphase, telophase, and cytogenesis second. Then anaphase second. So this is the chromosome blue color and also red color of the X and Y chromosomes are then changes and doubled in number to form a single cell to double cells to 4 or tetrads. These are the reactions. So this is also grand scheme. The mitosis is also 2n stages. So these 2n stages, then same stages of chromosomes, same identically similar, does not change as a chromosome number. That is called a mitosis. Here meiosis 2n, but the 2n is which divides the two meiotic divisions. First meiotic divisions, the same chromosome number of states of nucleus or chromosome. Then second stage, the double number of chromosomes are divided to the single number of chromosomes of two cells. Means one 2n cells. Also formation of the 4 cells. So this is also meiosis of the ground plan. That is also got a parental cell. Then the 4 cells are formation. So this comparison of mitosis and meiosis is based on the figures. So this is a mitosis, is a prophase, metaphase, anaphase, telophase. But meiosis, the meiosis are then also got a meiosis 1 or meiosis 2. The two types. The meiosis 1, prophase, metaphase, anaphase, and telophase. Then meiosis 2, then also called the prophase, metaphase, anaphase. There are 4 daughter cells formation in the last, that is called a comparison of mitosis and meiosis. Differentiations are there. This one, but this is also the total meiosis, which can takes place in the different stages of the chromosomes. First phase, prophase, the metaphase. So this is also telophase, also anaphase. The 4 stages, which is seeing these figures, which is then the total nuclear structures. Then the spermatogenesis and oogenesis, the 2 type of the spores inside of the formation. So this is important of the figures or a chromosome structurally. It can be changes or middle stages and peripheral parts. The chromosomes, pair of chromosomes are multiplied to the double pairs. Then double pair of chromosomes are triple pairs. That is. So this is a spermatogenesis. In the spermatogenesis, the mitosis also meiosis are here. So then the mitosis, autosomes and 6 chromosomes are there. Our autosomes are called A, and also 6 chromosomes are Y. These Y chromosomes are A, X, and A, Y. Both are present. Meiosis 1, the 2 cells are formed. Meiosis 2, then meiosis 2, only 2 cells. Then it is also got applied and applied. But here applied cells are only deployed in starting stages of the meiosis 1. Meiosis 1, after end of the meiosis 1, 2 cells. These 2 cells further divides to the meiosis 2. It can formation of the 4 cells. Each meiosis applied cells reduce are reducing to form of the two cells. That is the total, the four cells formation in the meiosis 1 and also called a meiosis 2. So this is also character. Then meiosis, then it is also process of meiosis, cell division that offs the chromosome number, creates a gametes for sexual reproduction. It is also mother and also father. Then process of the in the mother womb also got a father is also sperm. The fetal is also got a child, also zygote. That is then the laboratory, the mitosis and meiosis are differentiation here. So this differentiation is also in the mitosis and meiosis only. Synapses are differentiation. The synapses, the chromosomes, which can be fused to the peripheral parts of the two sister chromosomes. Then the chiasma takes place. During the chiasmas, one pair of the part is shifted to the other pair. Then other pair of the shift chromosomes comes from the first pair. This is also chiasma, also sigma. This is important of the two daughter cells are formation. But what is the means of the meiosis? The term meiosis was the coined by the J.B. Farmer in 1995. These types of division found in an organism in which there is a sexual reproduction. The term as the meiosis means the Greek word meum means diminish or reduce. Diminish or also reduces. The cells that undergo meiosis are called the meiocytes. So what is the meiocytes means? The sporogenous cells or sporogenous tissue which can ready to undergo meiosis. That cells is called a meiocytes. Three important processes that occur during the meiosis are pairing of homologous chromosomes. First one, synapsis. Pairing means the X chromosome and Y chromosome both are combined to pair. So then the pair is then X-shaped part formation. That X-shaped part means the first part of the chromosome shifted to the second to first. Then second important formation of the chiasma and crossing over. Chiasma means joined to each other. Then crossing over means the part is shifted to the first to the second to the first parts. Then segregation of homologous chromosomes. The segregation is important. The three important role in meiosis. Meiosis 1 or meiosis 2. So these are some few scientists are reporting the J.B. Farmer. Other scientist is also called this type of the year. Then meiosis was the first discovered that describes in urchin in animal cells. But here is this type of the scientist, which is this is also J.B. Farmer in 1905. That is also differentiations. Then, so the meiotic cell cycle involves the following stages. Then the interface is also different in mitotic and meiotic both. The interface meiosis starts after an interface, which is not very different from that of an intermitotic interface. Means during the pre-meiotic interface DNA duplication. Pre means also first time. So between the two meiosis reactions are between stages are so pre-meiotic divisions. It occurs during the S phase. S phase 1, meiosis 1. S means the synthetic phase. 1 also called meiosis 1. Then the prophase in the interphase which have a S phase anemia or synthetic phase and meiosis 1. The two stages are there in the interphase. Only first interphase, first meiotic division. The first meiotic divisions which can classify the S phase and meiosis 1. Then meiosis 1 is also followed to the meiosis 2. Then meiosis 1 in the interphase, yes, interphase means that is also S phase means ready to the synthesis of DNA molecules. Then in the meiosis 1, which can commonly Then prophase, metaphase, anaphase, telophase. In the prophase, leptotin, zygotin, pygitin, diplotin, diakinsis, the sub-stages of the prophase, which is important for the chiasma and chiasma and shifting of the chromosomes. That is also important. Then the prophase one, it is a very long duration and is also very complex. It has been divided into the following substages. First one, leptotin, zygotin, pygitin, diplotin, five substages, which major important in meiosis one. That is a long, that are more time taken to the chromosomes are, which can conversion to the changes of parental characters to daughter generation. That is leptotin or leptonema. Then chromosomes at this stage appears a long thread-like structures, elongated thread-like structures. That is called that really chromosome-like structures, not a chromatid. Then that are loosely invertebrate or interwoven. In some species, these chromosomes are bead-like structures. Beaded means round parts, which can be that round part is based to shorten sizes. Then round part is also dissolved, that the long or elongated and linear type of the model of the chromosomes appears in the metaphase. So then that are loosely interwoven. In some species on these chromosomes, bead-like structures called the chromomers are found all along the length of the chromosomes. Chromomers means round shape. So this meiotic division is which can take place in an anther. That is important. In the female gamete enzyme is called the and gynoecium or ovary. Gynoecium means ovary style stigma. Then ovary inside, which can take place in to formation of the ova or egg. But here anther is present in the male gamete enzyme. This anther, which has also present in the microspore mother cell. Every microspore mother cell undergoes reduction division. Means the sperms or male gametes formation is definitely undergoes reduction division. That in the leptotin stage or any stages of the meiosis, which can take place in the anther and ovary in plants. Animals is the testis and ovaries. That is also differentiation of the plants and animals. So then in some species, the chromomers are bead-like structure called the chromomers are found all along the length of the chromosomes. The length of the chromosomes are elongated, and so this is also got a prophase. You see that the prophase is a prophase inside which have chromosomes are then which can conversion, prophase, metaphase, anaphase, telophase, and cytokinesis. So these are all is also important, which can occurs inside of the cell of sporogenous tissue or anther or ovary, anther or ovary. This is also prophase. The prophase, first time, the prophase, which divides the 5 stages. Leptotin. In the leptotin, the chromosomes are red color and blue color parts are there. So the red and blue color is also 2 different parts of male and female gametes. Also got X and Y chromosomes. But here, zygotin. So these two, a pair of the X and a pair of the Y chromosomes are attached to each other. That is called the synapsis takes place. Synapsis. So in the pygitin, these synapses are which can between the X-shaped chiasma for chiasma. In the diplotin, these chiasma which formation to liberate to each other of the chromosomes. So in the diakinsis, the blue color part of the first chromosomes have red color small part. Then red color part of the chromosomes which have small part of the blue color. That blue and red color is transmission. to the X and Y chromosomes. It can changes of parental to daughter generation. That part is important for variation, mutation, and also got a genetical variability in plants. That so this type of the entire chromosomes are fusion. A part of the chromosome fusion is important. The entire of the chromosome X is fused to the Y. The total percentage more than 50% is a variable. A part of the small which transfer 20% of different to the parental characters to the daughter generation. So the here is also got a figures. Then leptodine, zygodine, pegytine, diploidine, and diakinsis, which then so this is also got a metaphase 1, metaphase 1, anaphase 1, metaphase 2, anaphase 2. That is also differentiations. So these are also the chromosomes and also sister chromatids also got a spindle shaped structures, which has a differentiations are there. So these differentiation is which are clearly then pegytine, zygodine are second part. What is the means of zygodine? The zygodine also got a zygonema. It is a characterized by the pairing of the homologous chromosomes. Also got a synapses, which form a bivalent. Bivalent means a pair of chromosomes to attach to the chromatid parts of one chromosome to the other chromosomes. That is also bivalent. The paired homologous chromosomes are joined by protein containing framework known as synaptonemal complex. Synaptonemal complex, the bivalent have 4 strands. That is, then it can attaches of the 2 different strands of the chromosome joined by protein containing framework. So this protein is important for the role of the chromosome construction. So these 2 different chromosomal parts are attaches. That is called a synaptonemal complex. The bivalent have 4 strands. 4 strands. So this is also end of the protein. That is also part of the sister chromatid. Here is also leptodine, this one. The blue color here is also zygote. And zygodine, the sisters of a chromosome. Zygodine, pegytine here. Then diploidine, then diakinsis. Diploidine or diakinsis, that is also different. Then pegytine. So our pegytine also got a pegynema. Pegynema. The chromosomes appears as a thickened thread-like structures. So after attaches to the chromosome parts, which are pairs, 2 bivalent pairs, which thread-like structures. At this stage, exchanges of segment between non-sister chromatid of homologous chromosome known as crossing over. This example. The crossing over is a non-sister chromosome. Non-sister means not XX, same pair of chromosomes of living organism male, but one male X, one female X, 2 are still sister A, then also non-sister. Non-sister. The same type of the XX chromosome, example female, but that is got sister chromosomes. So male X, female X, that is got non-sister chromatids. So these non-sister chromatid homologous chromosomes are known as a crossing over occurs. During the crossing over, only one chromatid from each of the 2 homologous chromosomes takes part. The nucleolus are still persistent. So this is a important of the pegytine. Then pegytine. So these are pegytine of the proteins. This is also pegytine. Means then blue color and red color both are different. The blue color may be X, also got a Y color may be Y chromosome. Homologous chromosomes, centromeres, and kind of more synaptonemal complex here. This is also synaptonemal. The blue color and red color total chromatid parts are joined. That is synaptonemal complex. But here is a sister chromatids. The blue color 2, then also red color 2. Sister. Non-sister means red color 1, blue color 1. That is called non-sister chromatids. Sister chromatids and non-sister chromatids are joined. That is also then pegytine. So, but then the pegytine stages, which important further. So this is also synapsis pairing of homologous chromosomes. Here is a parental, here maternal. Then crossing over will occur here. Oh, this one. A. Oh, this is also called capital A and small a. Here is a small a and a capital A. This part is also, you see, chiasma takes place or crossing over this part. Crossing over, then after crossing over, the A and capital A, small a and capital A, there are differentiations are there. So this is a total the red color and also light red color. This type, the synapses are pairing of the chromosome, then also diplotin or diplonema. First one, leptotin, zygotin, pagetin, diplotin. Then also third or fourth stages. At this stage, further thickening and shortening of chromosome takes place. Homologous chromosomes are start separating from one another. So after fusion, after chiasma takes place, the homologous chromosomes are separate to each other. That is called diplo. Diplo means two. Then isolated or separated. That is also called tin, also called nema. Separating from one another. Separation starts at the centromeres and travel towards the ends. Centromeres, center part to the peripheral part. Then the center part of the centromere jointing. Then also chromatic parts are related. Some type of the chromosomes are fusion to the peripheral and middle part are entire. The entire synapses are peripheral and also middle part. The entire is also more variations. So the homologous chromosomes are held together only at a certain point along the length. This is important. The homologous certain length, the length also got such points of contact are known as a chiasmata. Which part is attaches, which part is a starting of the attachment of non-sister chromatids, that is called a chiasmata, and represent the places of crossing over. The process of terminalization is completed in this stage. Terminal to middle. Terminal means in the tip of the chromosomes. The centromere is the middle part of the chromosome, medial to terminal separate. That is also terminalization. But here is also called diplotin. Diplotin chromosomes are structures. But here, assembly of a diesters of a synaptiminal complex. Then synaptiminal complex here, the parental sister chromatids. Here is a leptotin, homologous pairs. Then here, pagetin, full formed complex. Then here is also diplotin, but here is also zygotin. But this is also different stages. Then synaptimal complex, outer formation. That is a total described in this figure. So this is also the interface and homologous chromosomes, meiosis 1 and also meiosis 2 are there. But in the diakinesis, the chromosomes are continue to undergo further contraction. The bivalent appears as round, darkly stained bodies, and they are evenly distributed throughout the cell. Then the nuclear membrane and nucleolus disappears. This is the end of the prophase. In the prophase, first one, leptotin, zygotin, pagetin, diplotin, diakinesis. Diakinesis and diplotin both are termination in separation. Leptotin, zygotin, pagetin is a three divisions. Subdivisions are important of chiasma formation and genetical variability, genetical recombinant process occurs in three substages only. That is the nuclear membrane and also got a nucleolus also disappears. So this is also then the leptotin, zygotin, pagetin, diplotin, diakinesis, all 5 stages which are clearly seen in these figures. Then, so this is also autophases of the two diploid, also red color and blue color. The red color chromosome and blue color chromosomes are only terminal parts is attached. See that terminal to centromeres. That part is jointing, which is a important of a that fusion to the chiasma formation and also called a synapses. After synapsis, X-shaped chiasma formation. This X-shaped chiasma, which is responsible to the variability. Then this variability up to millions of years is called evolution. This evolution, which occurs from the unicellular to multicellular organisms. This is important reactions, important process for how to develop the man, how to origin of the man, chimpanzee, monkey, gorilla, which are there. Then also this type of the chimpanzee comes from the gorilla comes from the monkey. That is also process here. This. But then proof is one. Here is also complete. But this is also crossing over another. Crossing over. Chromosomes are crossing. But this crossing is here seen that bandage parts. These bandage parts are then green color and also blue color, sky color part attaches in that. Crossing over. But here the crossing over is also. The above one is P1 or M1, A1, T1, P2, M2, A2. That is also generations. Here, crossing over major important for the meiotic division. The meiotic division is important role only left out in zygote PGT. The 3 is a major. Then also metaphase 1. So this all is also called a prophase 1. But in the metaphase, the chromosomes are most condensed and have smooth outlines, which the centromeres of bivalent are connected to the poles through the spindle fibers. Spindle also fiber. Spindle means center part of the elongated parts, that is spindle. That the bivalent will migrates to the equator more. They disperse to the poles. The centromeres of the bivalent are arranged on either side of the equator, and this type of orientation is called the co-orientation process. The orientation means the middle part is elongated and the peripheral part is short. The arranged to the either side, the equator and the type of orientation. So this is a metaphase. The metaphase is a first metaphase. Then here is also but the metaphase 1. Then the PGT, diplotin, diagency, which are all stages are prophase. So but here prophase after anaphase. Anaphase, the chromosomes in a bivalent move to the opposite poles, disjunctions. Each chromosome passes 2 chromatids. The centromeres is the first to the move to the pole. Each pole has a applied number of chromosomes. This is a important of telophase. Then the nuclear membranes are formed around the groups of a chromosomes at the 2 poles. The nucleus and nucleolus are recognized. The same to the mitosis, but only prophase, metaphase. Prophase is a difference or most difference to the mitosis and meiosis. But here is the meiosis 2. Then in the meiosis 1, then the prophase, metaphase, anaphase, telophase. But in the meiosis 2, the second mitotic division is similar to the mitotic division. Second meiosis is similar to the mitotic divisions. First meiosis is different to the first mitotic divisions. That is differentiations. The first meiosis is more differentiation. The second meiosis is similar to the mitosis. In the mitosis, 2 equal cells, but here meiosis 2 equals to double or 4 cells. First meiosis 2 cells. In the second meiosis, the 2 cells are doubled in the 4 cells. That is also 4 tetrads formation after completion of the meiosis 2. Meiosis 1, meiosis 2, both are important for the forces formations. In the meiosis 2, in the first meiosis 2, the prophase 2, or metaphase 2, anaphase, or telophase are any 2. So then the prophase 2, the chromosomes are condensed again. So the nucleolus and nuclear membranes are disappears. The chromosomes are two chromatids, each become a short and also called a thick. Uh, the prophase 2 is similar to the mitotic 1, mitosis division. It is a condensed again. The nucleolus and nuclear membrane is also disappears. So the chromosomes, two chromatids are each become short and thick. Short and thick, that is also similar to the mitotic division. In the metaphase 2, then spindle fibrous, it can appears in the, and the chromosomes get arranged on the equatorial plane. Auto orientation. This plane is at right angle to the equatorial zones. Right angle to the equatorial plane of the first meiotic divisions. First meiotic divisions and the second meiotic divisions are, it can be right to the metaphase 2 or metaphase 1. But here is also meiosis 2. But this type are prophase 2. Then the chromosomes are prophase 2, that the movement, it is a movement is occurs. But meiosis 2, prophase 2, the meiosis prophase 2, the chromosomes are comes to the middle one, middles, and also anaphase, and also telophase. This is also differentiations of a. Then the meiosis 1, it is also, then anaphase 2, each centromers divides and separated the two chromatids which move towards the opposite poles. So the metaphase, the equatorial plate is there. The equatorial plate have a pairs of chromosomes are there. So these pairs of chromosomes are passed to the north pole and south pole, polar region. That is anaphase. Ana means the two chromatids which have moved towards the opposite poles. In the telophase 2, the chromatids move to the opposite poles. The nuclear envelope and nucleolus reappears. Then the prophase 2, the nucleolus and nuclear membranes are disappear, but it also reappears. Thus, at each pole, there is a reorganization of the applied nucleus. So, after anaphase, then the cytokinesis means cytoplasms are divided two equally. The cytokinesis are the division of cytoplasm takes place by cell plate method in plants and any furrows methods in animals. The cytokinesis may take place after meiosis 1 and meiosis 2 separately, or sometimes may take place at the end of the meiosis 2 only. Means the cytokinesis are separated to equals. Then the cytoplasms are separated in two cells of meiosis 2 and other two cells of meiosis 2, both are four cells formation. This is also, then here is also the total part of the, what are the differentiation of the meiosis 1 and meiosis 2. This is important. The total structure of the meiosis are here. Type 1 features, type of the division, meiosis 1 reduction division, but meiosis 2 equational division. Reduction, equational differentiations. So the reduction means reduce the number of chromosomes. The reduced number of chromosomes are doubled in equatorial divisions. Equatorial divisions.